So the program of this IPE, first I will present the 5 pillars of IPM, remind you. Second, I will present the project and the assessment tools we use in the project. After that, we will see 3 examples from Netherlands with Alfred Hunting that will present holistic integrated crop management systems and some examples in cabbage and wheat. After that, Luc Kruijmans will present strip cropping tested in the Netherlands. And late, we will see Isaac from Denmark that will present the experiments in wheat about cultivar mixtures, but also the combination with biocontrol. The fourth part is a discussion with participants, but it will be without voices, but with the tool Mentimeter. And late, I will present the upcoming events of ADOPT-IPM for the last year of the project, because we will finish in November this year. I forgot to present me. I am Philippe Delval. I'm a technical engineer for an applied research institute in France, and I'm based in Lyon. So I use the IPM definition that are inside the directive, the SUD directive, and you can see the main topics about IPM is that we have all available plant protection methods, and of course we have to choose prioritize some methods. We have the levels or thresholds we have to take care. We have to justify economically and environmentally, and we will see in the tool that we use these criteria, and minimize risk to human health and environment. The objective of IPM is to promote healthy crops and use maximum of natural mechanisms. In another project called IPM Works, we define 5 pillars for holistic IPM to provide a healthy crop. The first is landscapes with a lot of solutions. The second one is to design cropping systems with some agronomic solutions, but also genetic. The third one is to prefer non-chemical options like biocontrol, mechanical weeding, and so on. The fourth one is to optimize decision-making to avoid unnecessary treatments. And the last one is to increase the efficiency of the treatments with pesticides, but also biocontrol products. So today, the 3 presentations, the first one will be a more holistic approach on ICM with Alfred. The second one will be an example of use of strips. And the third one is the combination between cultivars, genetic, and biocontrol products. In ADOPT-IPM, we have 5 work packages that interact, of course. In blue, you can see that we have 2 research work packages on optimization of existing methods, but also on development of innovations. We have the demonstration and all the trials you can see are in the demonstration trials, and we have to valorize this. But we have in leak with the support tools and assessment to evaluate the IPM packages. These IPM packages are designed by the people from each country with adapted solutions to the context, pest, soil, climate. And at last, we have dissemination and communication tools. We will talk about this at the end of this webinar. For instance, for wheat, in optimization of existing methods, we have strips, use of beneficials, micro BCA, study on agronomy varieties, compost solutions. And in development of innovations, we have a lot of study about biological control, DSC, are dark septate root endophytes that are fungi that can promote plant stress resistance, and with the objective of suppression of Fusarium wilt. And we test also new biostimulants and elicitors to use on Septoria, for instance, in the fields. We have, like you see in the map on the right, trial network. You can see that in the north we have more trials on wheat and on leafy vegetables, from Dijon in Rae, Jean Blou in Belgium, Wageningen and Aarhus University represented today. But also in Netherlands, tomato, with another colleague of Wageningen. And in the south, you can see that the people of France, Italy, and Spain are more involved on vegetables, tomato, in field and in glasshouses, but also leafy vegetables. And in China, we have also a network. You cannot see it here, but we have some trials on maize, for instance, because there are some innovations tested on a new pest, fall armyworm, that are now in Europe. That is in Europe, in the eastern part and in the Canary Island. I will present now the package performance assessment tool. Remember, take care, because you will have some questions into the Mentimeter tool after that. It's a presentation that comes from our colleague from Imperial College in UK and Aarhus University in Denmark. I present this, and if you have some question, don't hesitate to put it in the chat. So the objective of this tool is to compare the relative impact of two crop management packages, two or more, because in the trials, demonstration trials, we have the possibility to have at least two packages, but we can put more if we want. I remind that the package is a combination of practices or technologies, but it's not a receipt. It's adapted locally regarding the pest, the climate, the soil you have. So it's very important to remember that, because it's not we don't do the same things everywhere in Europe and China, but we adapt the packages with the problem we have everywhere on the fields. And sources for the tomato. We have two pieces to assess. At the left, a quantitative assessment about impacts of pesticides on human health and environment. And on the right, qualitative from the experts that do the trials. So we have in this tool a part that is based on database, and one part, the qualitative one, about the experience of the experts. So for the quantitative part, the objective is to measure the impacts of pesticides on human health and environment, and we calculate a pesticide load index. This index is based on the work that the Danish people from Aarhus University done before, with the calculation of chemical properties to represent potential human health, environmental toxicity, and environmental persistence loads. And the value is between 0 and 1. And for this, we use the PPDB based to qualify all the pesticides used, and each part represents 1/3 with different number of subcategories. For instance, for human health, you have 7 subcategories. For becoming in the environment, 5, and for ecotoxicity, we have 15 based on aquatic and terrestrial impacts. You have an example of this index for glyphosate that is around 0.21, and we have all the part of this calculation on the radar on the right. For the qualitative part, we have an assessment from experts. So we have 6 indicators, and for each indicator, the experts have to qualify the value of the package and the confidence in that value. And at the end, the objective is to combine the 6 indicators into one only to summarize all this. You will see that after. So for these 6 indicators, you have alpha represented by crop losses, the production, the productivity. You have another quarter for the grower, the direct cost, time management, and the usability. But it's evaluated by the experimentators, and at the end, the classification for environment and human health and safety. The objective of this last part is not to represent direct impact, because you saw that we use a pesticide load index before, but it's to represent the opinion of the experimentator about the use of each package around environment and human health and safety for the sprayer people. So for each indicator, we use the same system. You have on the left the rating by the experimentator, and on the right, the confidence. So you have 5 classes for rating and 4 for confidence. So, for instance, you have 2 examples here. If the experimentator classes the rating acceptable and the confidence very high, you can see that on the graphic you have a very high number 4 that represents the acceptable rating and the confidence. But if the confidence is low, you will have another distribution around the different classes. It's the same if you use a rating not acceptable, but a confidence very high, you will have the number 1 at the left representing the non-acceptable rating. So for each package, you will have a comparison between the 2 or more packages tested into the demonstration trials with the 6 indicators. So we can have at the end the performance, but the performance with the ID of the IDs or the experience of the trial experimentator. It's why the 2 last indicator, environmental and health, are more representing the ideas about these points, but you saw that the pesticide load index have a very important a very great importance at the end. So we can, at the end, compare the impacts of the 2 packages from at the left one and the second one, and to have an idea of the performance. You can see that on the right, you have the combination of all the indicators to form only one performance indicator with the utility that represents the confidence, the sum of confidence for each indicator. So in summary, this performance assessment tool uses a weighted 6 indicator assessment that includes a entity to produce a single graphical output, like you see, and we have to add after that the pesticide load index to have the complete evaluation of the packages. The contacts are in UK, John and Adrian, and Virginia is based in Denmark with Isaac in Aarhus University. All these results will be in portal developed by CABI, the pest management portal. It's a practical double tool where you can find IPM options, selecting your country and your crop, but also view the performance data of all the trials that are in our demonstration network. So at this moment, we have only results in lettuce in Italy and France, and in tomato in Spain, Italy, and Italy. But at the end of the project, the idea, the objective is to have all these results available directly on the portal. At the end, I will send you all the information to go to this portal. So the contacts are Cabby people. Chris, Catherine are in UK and Fang in China, and they work on this portal to make available all the results of the project. So I finished my presentation. So I will stop my sharing my screen, and I will do the floor to Wilfred. It's okay for you?
Yeah, you want to move right on? Fine. Share my screen. Okay. Yeah, it's visible.
Yeah.
All right. So I labeled the overarching topic here integrated crop management because that's a concept that we have been developing and working on over the past, well, 5 to 10 years, somewhere like that. Actually, we first started doing things and then thought, well, what are we actually doing? And that turned out to be the integrated crop management approach. And I'm going to share some work that we did inside this project and outside this project in the past few years. Some of it. So why integrated crop management? What is it in relation to IPM? Well, this definition of integrated pest management by the FAO was already in Philip's presentation, so I don't need to go into detail there. But it has components of consideration of integration, but also economic justification. So there's a lot there. So not a bad word about that. But then if you go to practice, and we all know this IPM triangle or variations of it, then this is more or less the theory. And in common practice, this in many cases is practice. So all these basic building blocks go to the background, and a lot of the daily interest is about pesticides, how to use them, when to use them, what are their effects, how can they be combined, stuff like that. And of course, there is some monitoring there, but if the basic approach is to use pesticides, then you have a lot of you have a whole other concept in mind than when you have to do without, as a in contrast. So that started our thinking, and we thought, well, we may need to reshuffle all these components, and that's how we came up with this concept of integrated crop management, in which we define 5 pillars. We label crop diversity, that's on the top, as the first one here, but you might also argue that monitoring and evaluation is the first one, because first you have to know what your growing circumstances are before you can make any decisions. You will make different decisions when you're in mid-Denmark than when you are in Spain. So, but then based on your situation, you can start designing or adjusting a cropping system. And that has to do with rotation, but it also has to do with cultivation types like strip cropping or pixel farming or plot size, all stuff like that. They're all have their influence. And then based upon that, you start rethinking maybe cultivar choice instead of going for the maximum yield and with a lot of fertilizer and pesticide input. When you have to do without or you have to do with less, you make different choices. And the same goes for what are the influence of soil type, soil quality, soil condition, if you will. And also, of course, targeted control is part of your approach. and then you end with monitoring and evaluation again, or then that closes the circle. And maybe one extra layer, so to say, in contrast to IPM, is that we have tried to categorize certain measures or certain components in the approach. So we distinguish prevention of buildup of, weeds, pest diseases, or any possible attack to the crop, so to say. And then another component is to try and create as strong crops as possible, or maybe not crops or plants or cropping systems, key element that you work on strengthening. And then this targeted control, so this old school using pesticides is more of a closing part, not so much the focus of attention. But you will have to do maybe stuff to try to limit expansion of any weeds, pests, and diseases. And we also recognize a fourth element, which is the supportive tactic. So that is scouting, that is the support systems, that is also knowing maybe the history of a field, stuff like that. And the combinations make the approach strong if all goes well, and then you have also some, let's say, risk mitigation in place. So that works out like this. I'm not going into detail there. I think I touched upon the key elements, and we call it integrated crop management. And depending on the situation and maybe also the boundaries or goals that you might have, you put certain things more upfront in your decision making than in other situations. So you could label any integrated whatever management there. The key element is that you start with your situation and start with your goals, and then say, okay, what is priority and what is less important. So this is more or less the concept that we work on in short, and then go into, let's say, crop specific situations. And in our situation, that was working on cabbage. So we labeled that integrated cabbage management, which, might lead you to the understanding that we have included all the cabbage management here. That's not the situation in the work that I present here, but it's always there. So this, what I forgot to say is that this concept of integrated crop management is basically, it's like a checklist, and it makes sure that you will never overlook stuff, rather than a blueprint of how to go about. So as I said, depending on the situation, and the situation can be defined in many ways, you make different choices. So, and it also has to do with who can make the choices, and what are the risks that are there, what is the risk perception, which is maybe even more important, and maybe also if you have certain alternatives, how practice ready are they? So you can think of a lot of options behind your desk, but then going into practice and make it work, that's really where the challenge often is. In part of our work, we talk to growers first, and then they say, we ask, well, if you grow your head cabbage, what is your top priority? And they said, well, it's onion thrips, and then onion thrips and onion thrips, so to say. So okay, that was clear to us. We have to do something there. And obviously, that is a prioritization based on the current situation, because currently growers conventional growers say, well, we have insecticides and fungicides and any pesticide for certain situations, and then for some we don't. In this case, thrips. So then that's top priority. Whereas if they would have no pesticides at all, or that would be different. that would lead to different prioritizations. So it depends on your starting point there, but for us this was a given, and that also trickled down in the work that we did within the ADOPT-IPM project. So, and with this, I'm showing some experiment data, not all of it, but we touched upon quite some aspects as a part of an integrated crop management strategy. So we did something with cultivar choice. We did work with planting date of the crop. We did some work on companion crop. We did include some tillage elements in our work. We worked with cover crops, and we did some work on biocontrol. And in any case, this quality, yield quantity and quality is basically your closing parameter, because if you make a different choice, but you suffer poor quality or quantity, that will lead, well, that eventually that will bring you out of business. So that's an important parameter. So I'm showing first some data of the trials that we did within the project. So we did one trial in 23 and two trials in 2024, in which we compared two planting dates. So let's say a standard date and a delayed planting date, because delayed planting it has a reducing impact on thrips damage, is the assumption. We work with two cultivars who have cultivar sensitivity within the crop, and on top of that we had three, let's say, spraying schemes, you might say, although spraying schemes is not the correct term, because we also included an untreated control there. So in all we had, and they were all mixed, so we had 12 treatments in the trial, and we ran that in 3 pods. And I'm showing some thrip damage data at harvest. You see some picture there of what the damage that thrips caused, because they caused these the tumor sensors in the leaf. So it's really a plant reaction that is seen as, it's not a yield quantity issue, it's basically a quality issue, and the market demands visually clean cabbages when sold. So in practice, cabbage growers will peel as much leaves off until they have a clean cabbage left. And of course, that's a kilogram loss, but it's also work. And we use a scale in which we have a categorization on a 0 to 6 scale per leaf, and the deeper the leaf is into the cabbage, the more weight we give it. Then some data. So we did 2 cultivars in both years, by the way. Colmar, which is a relatively sensitive cultivar, and Lucas, which is relatively resistant. Well, that's clearly visible also in the left graph. But the assumption that later planting would also lead to less damage by thrips was not visible. Strikingly, I could say. So that's the left graph, and in the right you see the treatment effect. So we saw that, let's say, spraying helped, and that's, well, should not come as a surprise. So that's a trial in 23. This is a result of a trial in 2024. And you saw in the previous slide, you saw an interaction graph. in this trial we didn't have interaction, so I'm not showing that. So we saw again the cultivar effect, that was already visible in the first year. But here we also saw that later planting resulted in a lower thrips damage index, which is according to hypothesis. But then in this trial we didn't see very clear spring schedule effects. This work is still under analysis. That is, we haven't not finalized report, so we didn't have a chance so far to have an overarching analysis. But it's clear, at least, that the cultivar effect is there, and we have to find out what was happening with the planting dates, and also with the spraying strategy, because that's probably in the details of what was used when and where to explain this. Then outside the project, we have a public-private partnership running, in which we also did some work. I'm showing 3 trials that we did last year, and in these trials we already selected the late planting, so late planting and a robust cultivar. So there's no comparison in these trials; it's more or less a given. And we did some work with clover as a companion crop. We have applied this in different ways. The idea is, and so as far as I know, that was never tested in cabbage crop before with specifically this goal. Although it has been tested in leeks, so clover with leeks, but not with cabbage. The assumption is that it will reduce thrips damage, and we're not completely clear what would be the mechanism behind that. Maybe Luke has some insight there. We'll see later on. And as we did this as a strategy approach, not so much comparing all the elements, although trying to say something about them based on the results. So we also combine this with different control strategies. And one of the reasons there is that we did 2 of the trials in farmers' fields and really had a talk beforehand with the farmers on what level of risk they were ready to take. And so 2 trials we did with farmers. The third was more or less a demo, and that was done into strips that were already sown with clover. 2 types of clover, as you can see on the screen, and that was not a completely statistically justified approach because we had 2 strips, so we couldn't really replicate well. But we tried to make the best out of it, but it was more a demo than a trial in that perspective. So the clover was sown autumn before. You see in early spring the picture in the screen, and we had some weediness there. So to control the weeds as best as possible, we did weeding before planting and continued weeding also to let's say control the clover in the competition with the cabbage later on. We'll see that. So this is really an experimental system because we did strip cultivation. So that's really narrow strips of, let's say, 10-15 centimeters of power cultivation in which you plant your crop. And you can imagine that the clover is already established, so it has a head start. So it's really tough for the cabbage to get going. and we have done this with GPS systems, but then the practicality, well, results sometimes in systems that are not fully aligned. So you see in the right picture that we have a strip cultivated, but then with GPS planting afterwards and GPS not having the correct signal, stuff like that, then we have this plant at the edge of the strip instead of in the middle. So that was really some of the practicalities that we faced, and it underlines the aspect of risk perception. What you can think of behind the desk also has to be executable. I already said we continued mowing. We did this with a lawnmower for at least 4 times after planting the cabbage, until we thought, well, we harmed the cabbage too much, or we estimated that they would be ready to compete with a clover. So post planting, we did one herbicide application, of which we knew the clover would have some effect, but that's maybe partly intended. And then we started a thrips control strategy with biocontrol starting early August, basically the period that the cabbage starts forming the heads, because we think that before that, in contrast, by the way, to what some growers say, but we think that thrips control is unnecessary, or you might say useless before that. But then later in the season, this is more or less the picture. So, okay, you have the cabbage, it will survive, but it will not reach its full potential as a cabbage crop. I can spoil already. So we did, as I said, with this thrips index, we did the assessments at harvest, and also included cabbage weight, because there was some discussion with growers that smaller cabbages might be less favorable to thrips, so they could have less damage. So we did a correction there, and that results in this picture. So what I forgot to mention, that we had these 2 clover strips, but we also had, like, let's say, a conventional strip beside that. So that's the right third. So the no clover in the graph. You see in the gray bars, you see the cabbage weight on the right y-axis. So you see already that this cabbage clover cultivation led to a lower cabbage weight. And you see in blue, let's say the calculation with this thrips damage index, and in orange you see the correction for cabbage weight, which I think is justified. And we saw clearly that these cabbages in the clover had a lower thrips damage. So apparently there's something there. We don't know exactly what. Well, it might be microclimate. So because you have, well, really a crop of clover which has a humidity in which maybe the biocontrol, which was a entomopathogenic fungus, has maybe better chances of establishing in the crop. So that might have helped the efficacy of that product. That could be a reason, but again, it was a demo, it's not a trial, so we cannot be all too conclusive here. But there's something there. So this was a very experimental, and I might say impractical, at least from a grower's point of view, approach, but very insightful, I think. We did also 2 trials in practice and also on a practical scale, because we used big plots of 12 meters long and have a spray boom, so that's also 13, 14 meters wide. So really big to have, let's say, migration of thrips if that would occur, to have mitigated that as much as possible. And also to make it practical for the farmer, because the farmer did, at least in one trial, they did the spraying. So that is that was one of the reasons. You see here already that we planted 6 May, which is not early. I mean, early would be early April, so it's already delayed planting. But in the second location, we planted on June 3rd, which is much later still. And the reason for that is a choice of the farmer, because he is in an area where he is suffers a lot of thrips because of onion cultivation and also alfalfa in the past. So he deliberately chooses to plant late, extra late. Trial setup for the rest is more or less the same. We did again a biocontrol strategy starting more or less when the cabbage heading started. Just a picture, nothing to tell here, I guess. We did undersowing of the clover in the first location, planted on 6 May, early June, so after the last hoeing pass. So that's also a practical aspect, that if you want to do as much as possible mechanical weed control in your cabbage, you can until then not sow your clover between the rows. So we did sow between the rows after the last hoeing pass. And then in the left picture you see some clover seedlings already. So that was, well, within 8-9 days, very quickly. And then all through the season, well, we saw clover, and that's more or less the whole story. So even on the right side, that's, I think, late August, maybe in September, you do see clover, but let's say, well, I said to colleagues, I would be surprised to see what will happen there. But apparently, we'll see that there was some effect. This trial of which you saw the pictures. So we have an untreated crops regarding thrips starting early August. And there were, I think, maybe 3 sprays in practice. And then we had the clover undersown, in which there was no additional spray. And then we had a combination of both. Well, if this is true for many more replications, then it's really quite a breakthrough, I might say. Because I was actually surprised of how much reduction we saw with the clover. So and I don't know exactly what the mechanism is. That there might be a mechanism in the soil, but it might also be the behavior of the thrips itself. It might also be that there is some effect of predators, natural enemies. We didn't look at that, so it's just a first. It's actually a first with this approach. Then the second location, different location, so 20 kilometers up north, different circumstances, planted a month later. We saw an effect of biocontrol. That's interesting, but we didn't see an effect of clover. So that's why I was both happy and cautious with the previous graph. So this is really, I mean, the cabbages were harvested 3 months ago, so we really don't know exactly where we are now. But the statistics indicate that the results are, well, that's at least fairly sound, and that there's something there in the clover cabbage thrips interaction. Put it like that. So I would label it as first insights in that respect. To summarize, not to conclude, but really call it insights. We see, and that's also recognized in practice, that cultivar choice is important, and that delayed planting is helpful. That is already taken up again in practice as much as possible. And well, crop seems to have a perspective as a companion crop, and we don't know exactly how and what the sustainability of that is. What we do know, and that's not really a conclusion, but that biocontrol products need to be used consciously, and the reason why I put it down is if maybe based on the findings in the demo, in which we think that this humidity is a component, so that really stresses proper timing and finding the right circumstances to apply these products. And well, there might be all kinds of synergistic effects, although there might also be antagonistic effects. We don't know, so it's really in that perspective in quite an early stage. And with that, I would like to conclude. So Philippe, do I stop sharing? Is there time for questions? What is the approach?
There's no questions, so you can stop sharing. We will continue with Luke, and after that we will see if we have some questions at the end. So Luke.
Yes, I will share my screen. Are we all good, Philip?
Sorry. Yeah.
Yeah. Okay. Great.
Yeah.
Well, then I will continue a bit with the cabbages where Hilfred left off, and a bit of a disclaimer that I also don't really have an answer for his question that he had for me. I know it's for the cabbage root fly. I would have a theory why the clover would help with for trips. I'm also a bit lost, so I'm quite curious how that would work. I will focus a bit on strip cropping. A lot of work on cabbage again, but also some other crops as you'll see later. And I want to start with a bit of a definition, if my computer allows me. So this is a bit of an over-exaggeration because in the Netherlands and Denmark, I don't think we have this big of a fields and approaches. But what we still often see is monocultures of just one crop, and those, as Hilfred already mentioned, I think it's good to create a bit more diversity in the field to get a lot of ecological interactions happening. And why that is the case, I just wanted to illustrate with this life cycle of a surfeit fly in Dutch zweefvlieg. And their larvae are, of some of their species, are predators of aphids, so they're important for as natural enemies and for pest control. But these surfeit flies, they need different things throughout their life cycle, right? So their larvae, they eat aphids, so they need an aphid source. But the adults, they actually don't eat aphids, but they eat pollen or nectar from flowers. So within this landscape, they need both flowering plants, they need plants where the aphids are on, they need places where they can overwinter as well, where they can continue their life cycles. So they need more than just one crop. If you just have one crop with their aphids that they can protect, it's not enough. So what we kind of came up with or stole a little bit from ancient practices in China is strip cropping. And here you can see the fields in Lelystad in the Netherlands. And the idea of strip cropping is that you have relatively narrow strips of different crops next to each other. In this case, in this example, we have two different crops next to each other. and the strip width is usually as narrow as possible, because then we expect bigger ecological advantage as well. But you can adjust it to the machinery available. That makes it kind of practically implementable. This is what it looked like from a bit closer up the fields. Here you can see the cabbages in the middle and wheat on the sides. And this was a combination that we've been trying for some years and did a lot of research on. And as I said, we had 2 locations, so this is where they are. I think especially useful maybe for the Danish colleagues, so they know a little bit where we're looking at. Wageningen and Lelystad is where the experiments were. And then we had several different designs, but there's 3 that I will talk about in this talk. So we had a monoculture of just cabbage, no other crops in there. Then we had the strip cropping of 2 crops that you can see in the bottom right picture. So that was cabbage with wheat. And then we also had one where we had even bigger diversity. So we had 6 different crops, including cabbage and wheat, but also barley, pumpkin, potato, grass, clover, strips in between, and some other diversification in there. So this is really a much more diverse system than this strip cropping of 2 crops. So those were the 3 systems. And then what we did was also focus a little bit on the cabbages, the insects that come to these cabbages and the feed from them. We counted insects throughout the season, followed their populations, and then at the end, from the same cabbages, and you can see on the picture this small blue stick, that was to mark the exact cabbages that we've measured throughout the whole season. And then at the end, we would harvest them and we would weigh the cabbage head. We would also look at damage of the cabbages. We had a bit of a similar measuring system as that Hilfred was talking about. And the nice thing was that we then could link all these things together because we were constantly looking at the same cabbages. And what the nice thing then was that we could try to see what happens if there's more or less herbivores on that cabbage or pest insects, however you want to call them. And the nice results, I think I tried to summarize them in this figure on the right. So on the top, you can see the system diversity, and let's say on the left, it's the least diverse system, a monoculture, and then on the right, it's the most diverse system. So this strip cropping with 6 different crops. And then in the middle somewhere, there is the strip cropping with 2 crops. And what we saw was at the same time more herbivorous insects, so higher abundance and species richness of herbivorous insects. So it was a little bit counterintuitive for us because there's quite some literature out there that says that if you do some form of intercropping, the herbivore abundances go down, but we actually found the opposite. Same with Hilfred's work with the cover cropping. There also the herbivore abundances went down, or the trips abundances went down, or the damage. In our case, at least for the abundances, was not the case. But at the same time, we also saw a yield increase, so that was a bit weird. And what we found was when we analyzed how the insects and the yield kind of linked together, is that we saw a lower yield loss per trip and a lower yield loss per caterpillar that we found on there in this really diverse cropping system. So even though there were more herbivores, we believe that, for example, they were actually controlled by natural enemies over the season, and then they cannot do as much damage as they would otherwise do in a more simple system. So in a way, these systems create a bit more resilience in that system. So that's what I try to kind of visualize here a little bit. Well, then we also looked specifically at these natural enemies. So we looked specifically at parasitic wasps. So these are these wasps that parasitize caterpillars, lay their eggs in them, and then at some point the larvae come out. You can actually see that in the left bottom. It's a bit gruesome, but there you see a caterpillar that had these parasitic wasps in them, and the larvae are there coming out, and then they can fly away and parasitize other ones. And they're very important natural enemies in these agricultural systems. And what we saw there is if we go from monocultures to strip cropping, we see an increase in the parasitic rate, so the natural enemies are more effective. And then we saw another slight increase if you go to strip cropping with 6 crops instead of 2 crops. And the interesting thing, I don't show it here, but the interesting thing is that once we harvested the wheat and the cabbage was kind of by itself, we actually saw that didn't have a difference in parasitism rate anymore. So this wheat crop is important in a regard. There's something that the wheat is offering to the parasitic wasps that cabbage cannot offer, and that increases the parasitism rate there. I will share these slides as well. There's a link to a video here where we also explain that. It's in Dutch, but there's some subtitles there, and we really try to make it as easy to follow as possible what we found on these parasitic wasps. Well, then we've continued this research a bit. This is work from a PhD student, Misty, who's also working on ADOPT-IPM on the project. And she followed up a little bit on the work that I did, but also included the natural enemies in there. And she found a very interesting pattern where in strip cropping, and you can see at the top, I hope you can see the cursor, I'm not sure, but I'll also say it. So on the top, you'll see the yields of 2 different years, and she found that there was a difference, but if you look a bit better, you see that in both years, the yield of the cabbages was a bit lower in strip cropping than in monocultures. But at the same time, she also found lower abundances of herbivorous insects, so these different aphids, bladluizen in Dutch, but also of their natural enemies. So at the bottom, you see 3 different groups of natural enemies of these aphids. So everything was kind of decreasing. And when she looked then at the oats that were next to it, she actually saw a bit the opposite pattern. So there was a bit of a higher yield in oats and strip cropping, at least in one of the years. And there were also more aphids, and there were also more natural enemies there. And what we think was happening in this case is that the insects, they kind of follow where the resources are, so to say. So if the plants are bigger because they're doing better, then you also get more aphids because they go to healthy plants, and you also get more natural enemies because there are more aphids. So they're kind of following this system. So it was an interesting finding. And the reason we believe that there is this bit contrasting pattern is that probably oats are a bit the winners in this situation. They're a bit more competitive than cabbage, so they can actually take up more resources than they do in monocultures, whereas the cabbage is a bit a loser in this comparison, or in this combination. So to summarize a little bit those findings, what we see is that strip cropping seems to be beneficial for both the herbivores and the predators at the same time. I have some more results to show, but I didn't show them now. But I believe that it's helping both these herbivores, potential pest insects, and natural enemies. And in the end, the survival is reduced of the herbivores because there's more natural enemies, and that makes that there's not a big loss through these herbivores in strip cropping. Bit of a nuanced answer there. And then we were actually curious because now we did this research in these experimental farms. We were a bit limited in how many different places we could measure. The scale was relatively small, as you can see, was 3-meter wide strips. And then we had a bit of luck on our side because we saw that since 2019, the number of strip cropping farms has been increasing in the Netherlands, and you can see that a little bit on here. And a colleague of mine told me that it wasn't even complete in this picture because there were even more strip cropping farms. So it was quite nice. Well, you can also see it a little bit in this graph. So here you see the area of strip cropping in the Netherlands in hectares, and you can see that it's really kind of growing, well, exponentially maybe, since 2019. So we really see a big uptake in that. It's still relatively small areas. I recently calculated, I think it was 0.1% of the Dutch agricultural areas or arable crop areas. So it's not that it's very common in the Netherlands, but it is increasing nonetheless. And what we saw is that farmers haven't really chosen a clear setup that's best. So what you see in these 2 graphs, the top one is the strip widths that farmers are using, and you can see that there's not really a clear strip width that's preferred by farmers, or that at least in practice. We see one at 6 meters, but it's actually from one farmer that has a really huge part of its farm allocated to strip cropping. And for crop diversity, we do see a little bit more this kind of normal distribution, so to say. But even there, we see still quite some variation in how many crops farmers introduce in their strip cropping situation. So we're also a bit curious, what is actually best from a pest management perspective? What kind of strip widths or crop diversity? And then we set up this farm network research all across the Netherlands. And you can see on this map the different locations where we measured. And here we focused on potato and on cereals because these are two of the most grown crops in the Netherlands. So it was easy to find a lot of farms that grew them in strip cropping. And we had this kind of paired design. So each of these farms that you see, there was a strip cropping setup and a monoculture with the same crop. So we could really compare the difference between a monoculture and strip cropping setup. And per farm, they had the same farming system. So they were either conventional or organic agriculture, and they had a similar management as possible. We really tried to have a good comparison per farm and then see across farms if we can see some general patterns happening. And then we had 22 farms that we measured at. And as you can see, some were only cereals, some only potatoes that we could measure, but most of them we could measure both crops at. Well, then we measured, at least related to the insects and to pest management, we measured a couple of things. So we used sweep netting, so just going with this net through the field to see how many pest and natural enemy insects were in the field. We used pitfall traps, which are a way of capturing ground beetles, and ground beetles are important predators of, for example, slugs, certain pest insects, but also weed seeds. And we used roof tiles to look at slug abundances in the fields, also get a bit of an idea of how many slugs were there. And the slugs we only measured for the cereals, because in potato it's not a very big problem, and in cereals it could be. Well, and this was a bit of our design that we rolled out across all the farms. So we had this kind of standardized design. On the left, you can see how we did it in the monoculture. And then here you can see one sweep net transect and 2 pitfall traps, but this is times 3. So we had some replications per farm as well. And in the strip cropping, as you can see on the right, we measured both in the middle of the strips and on the edges of the strips, because it could be that certain effects are strongest at the edges where really the crops are next to each other, and we're curious if it would also happen in the middle and if there's this difference between that. Well, and then the first thing that I wanted to talk about is these slugs. So our idea was that probably what we also heard from farmers is that slugs usually come in from the edges. So at the edges, slugs can be more problematic. So we're expecting that in strip cropping, we might see more slugs because of this edge effect, but that there would also be more natural enemies to solve that. But actually, the hard truth is that for slugs, at least, there's more of them in strip cropping, and any effects of natural enemies didn't compensate that. We did also look at their natural enemies, and we did indeed find that they also increased in strip cropping, but the extent to which they increased was much lower than for the slugs. So they didn't manage to kill the slugs enough to get this reduction in there as well. So a bit of a slightly negative response there. But we also asked the farmers how they viewed the slug damage that they had between strip cropping and monocultures. And what they said is that from the 20 farms that we asked, only one of them said that he saw slightly higher damage of slugs in strip cropping, whereas the other 19 saw no difference in slug damages. So the damages at least are not that much higher. I must also say that the slug abundance that we caught were relatively low in most farms, which I found very weird because we measured in 2024. And if you remember, at least for the Dutch people, it was a very, very wet year. And slugs were all the time on the news, but still we didn't find that many. What we also asked them is if they found damage on other crops in their strip cropping, and there we did see hear from a couple of the farmers that they saw damage on, well, for example, pumpkin, cabbage, beans, sugar beets. So there is a bit of a risk with strip cropping and slugs. But if I conclude there, I'm going to skip a little bit forward. I would say they're not a major problem in strip cropping. So it's not that if you do strip cropping, you can expect in any case a lot of slugs. But if you are at a farm where slugs have been a problem in the past, then it's good to be aware of that, and that, yeah, that might be problematic in those cases. Then, as I said, we had this sweep netting. I think, if I remember correctly, this is the last bit. So we did the sweep netting both in potato and cereals. We walked transects of 50 meters. So you can imagine one measurement point is 1 meter wide and then 50 meters long. So quite a big area that we sampled. And in total, we walked 35 kilometers slamming this net through to the canopy. So quite a distance. And we did this to get a bit of an idea of the pests over there. So we identified the insects that we caught to family level mostly, and counted how many we got of them, so that we actually have an idea how that changes. And the nice thing was in this research that what I often see in research, that they focus really on one insect species or insect group, and we actually could do analyses on a lot of different groups. So I'm showing them here. You don't have to remember them for the exam, but I just wanted to show it that for all these species, we could separately look at how strip cropping affected them. So we don't only look at how many in total happened, but also for these separate groups, and to also see if there are species that are responding in different ways. And yeah. Well, then I'm going to go to the most difficult part, because we made some very complicated graphs, but I will walk you through it a little bit to make it easier. So this is a graph of the overall pattern that happened to all the herbivores. So if we merge all of the herbivores together, kind of what happened in their abundances. And what you can see on the top is these blue lines. They indicate what happened in cereals. The purple ones indicate what happened in potato. And then the darker ones are for comparing the middle, and the light one at the edge. And we have this 0 line. That's an important line, because if the results are on the right of that line, that means that there's more in strip cropping than in monocultures. If they're on the left, then there's more in monocultures than in strip cropping. And the interesting thing that we found, again, a bit counter to what a lot of literature, scientific literature says about intercropping, is that we actually saw more herbivorous insects, or pest insects, if you want to call them, in strip cropping than in monocultures, and specifically in potato is a bit more compared to monocultures, and always at the edges it was more than in the middle. So really at these edges is where they, there's more of them. And then we also looked at all the herbivores that I just showed you, and then this is a little bit the pattern that you see. So overall, if we look through this graph, you see that most of the lines are on the right side of that 0 line. So for most insects, we see that indeed in strip cropping there's more. But the interesting thing is that it's not always the case. So for example, for the cereal leaf beetle, which can be a pest in cereals, there was not an effect of strip cropping. So for that, it's not the case. It's the same with aphids, which are also mentioned a lot, both in potato and in cereals. They were only more abundant in the edges of strip cropping, but not in the middle. So yeah, I used a lot the word herbivorous insects here, because actually not all of these insects are known pest insects. They might eat from the crop, but they might never be there in really high populations that they're really damaging. And what we see is that these less common herbivorous insects, their abundances tend to increase, whereas these typical pest species, actually not always. An important side note there that I wanted to give is for the Colorado potato beetle, which can be very damaging in potato. And for that one, we clearly do see an increase in strip cropping. And we think that might be because they overwinter in the soil. And if you do strip cropping, the next year, your next potato is actually quite close by. Whereas if you're in a monoculture, you have a potato field there in year 1. In year 2, you're going to have crop rotation, and then it's going to be another field. And these Colorado beetles that come from the soil actually have to find a new field somewhere else. So we believe that that's the reason why for Colorado beetle, we do actually see an increase in strip cropping. Well, then I said also natural enemies. These are, again, the similar figures. Here, the overall pattern. So at the top, you can see the natural enemy richness. So how many different natural enemy species do we find? And there we see an increase. again in strip cropping, and below that, the all natural enemies indicates kind of the overall patterns of abundance of natural enemies, and there also we see an increase. So they follow the same trend as the herbivorous insects, and you can see that also for when we look at all the different species together, that most of them are on the right side of this zero line, so most of them are more common in strip cropping than in monocultures. Well, then we also wanted to know what happens kind of per herbivore. So if you look in, natural enemies, they tend to follow the herbivores, right? They go to wherever the food is, so to say. So this is a graph that I personally would have expected. So on the x-axis, on the bottom, you can see how many herbivorous insects there were, and on the left, you can see how many parasitic wasps there are, and there's actually a nice correlation between them. So you see if there's more more herbivores, there's also more natural enemies because they follow the herbivores. They want to go to where their food is. And for parasitic wasps, you actually didn't find an effect of strip cropping. So it doesn't matter which system, it's always this kind of positive correlation, but there's no change. However, for if we look at natural enemies, so more the predators of the herbivores, then for example, for here the aphids and their specific natural enemies, we saw that if the number of aphids increases, then we see a larger increase in natural enemies in strip cropping than in monocultures. So per individual aphid, there's more natural enemies in strip cropping than in monoculture. So that's at least kind of a nice positive effect. For me, that gives a bit of an impression that these systems are a bit more resilient against pest outbreaks. There's more natural enemies that can be there to maintain them. Well, then the last thing I mentioned, these different characteristics of strip cropping, for example, the width of the strips, the diversity, and these kind of things. We also looked at how that affected how effective strip cropping was in this pest management story. And then we actually see for the strip width that, between, let's say, 12 and 23 meters, we still have to look a bit more in depth in where exactly this point is. But somewhere there, you start to see that it responds in a similar way than a monoculture does. So if it's below 12 meters, you can expect an effect of strip cropping. But if you go beyond that, it becomes a bit more nuanced. And beyond 23 meters, it just starts to act a bit more like a monoculture does in this response. We also looked at crop diversity, so how many crops did farmers put in their strip crop arrangement, and there we didn't find an effect, so it didn't really matter how many crops were there. And we also looked at the difference between conventional and organic agriculture, and there we generally see that in conventional agriculture the patterns are stronger. So in conventional agriculture you see a stronger effect of strip cropping on both the herbivorous insects and the natural enemies. So in conclusion, as I said before, strip cropping does seem to be increasing the abundance of herbivores, but also the natural enemies, and the effect on natural enemies is even a bit stronger than on herbivores. The effects are the strongest when we have relatively narrow strip widths. And I want to end with that more herbivores is not necessarily more damage in the crop, also because of these natural enemies that are there. These ones I'm going to skip, I think, because of time. And then that was it. Thank you for listening. And then I'm going to stop sharing because otherwise I can't see anyone.
Thank you, Luc. Just question about your trials are in organic or conventional fields, and the distribution in Netherlands. You show the maps. Mm-hmm. What is the situation about this?
Yeah, so for the trials that I talked about at the start, the ones in Wageningen and Leefland, those were in organic conditions. Reason for that was that we really wanted to know what the insects do, so we didn't want to use pesticides because then you also don't really know how the insects are going to behave. For the farm network research, I think it was about 1/3 of the farms were organic and 2/3 were conventional farmers that were in there.
Okay,
and there we originally. Sorry?
No, you can continue.
I just wanted to say that what we tried was get kind of an equal spread. So we originally tried to go for like half of them organic, half of them conventional, but it's just the case that there's more conventional farmers. So it's more difficult to find. It was more difficult to find organic farmers that had strip cropping and a monoculture and have this whole thing. So there's quite some bottlenecks there. That's what yeah.
And do you have some data on trips with this technique, or?
With the sweep netting, not, because trips are a bit tiny, so it was difficult to really get them well out of the sweep net. For trips, sweep netting is not a great method. Usually you get this mass of plant matter in there as well, so then they're going to stick to the plant matter. It's quite difficult. So we looked at a slightly bigger insects. For the other work we have, so the work that I started, we also looked at what trips are doing. And there we did see that in strip cropping also there were slight increases of trips. It depended a bit per year. Trips were not a problem every year, and they were also not a problem in both locations every year. But I think if I remember the data correctly, the years that there were trips, they tended to be a bit more abundant in the strip cropping than in monocultures.
Okay, thank you, Luc. Now it's time to Isaac with the Danish trials, so you can share your screen.
Okay, can you see my screen?
Yes, it's okay, but
All
you
right,
can.
and I suppose you can hear me clearly. It will be in the presentation mode very soon. Okay, I guess now you can see it in presentation mode.
Yeah.
All right. So thanks for the earlier presenters. So there is one common theme that people were talking about, that is diversity. So Luke just showed about the role of strip cropping in crop pest management. And I will talk about something similar, but what I will talk about is called cultivar mixture. But the overall objective is the same, is to increase diversity in the field. And as we all know, diversity has a huge impact on crop pest populations. So my presentation focuses on cultivar mixtures and how it impacts septoria in winter wheat. And then I will show results about alternative products, in this case, sulfur, how we can integrate this with cultivar mixtures to control plant diseases. Now, just to start, to set the scene, I'll give a brief introduction about septoria. It's a foliar pathogen that is caused by a fungus called Zymotic septoria triticide. And these are typical symptoms on my right side here on the slide. You see this kind of necrotic lesion, which is surrounded by a yellow chlorotic area. And then you see this distinct black object on it, and that's actually a pycnidia. And these are responsible for secondary spread of the pathogen of the disease in the field. Septoria epidemics are generally very, very problematic. They cause massive yield losses, especially when the attack is severe during the green filling period. And during this period, under favorable conditions, you get about 2 tons, which translates to about 28 to 30% yield loss. So that is very significant. Now, the major means of controlling the disease, you can have a bit of host resistance, but the main means in Europe is that we use fungicides. However, most of the fungicides we use, mostly in the class of azoles, there are resistance against them. And some of them also are in this SDHI, succinate dehydrogenase inhibitors, and then the quinone outside inhibitors. Most of these fungicides, there are a lot of cases of resistance against these fungicides. The other problem with the fungicide is that we have withdrawal of fungicide. Recently in Denmark, PFAS fungicide are to be withdrawn from the market. Example is fluopiram, which is a major fungicide used against septoria. And a lot more fungicides have been delisted for other reasons. So overall, fungicides, although they are relevant, there is a challenge with their use because of resistance or regulations that is restricting their use or withdrawing their actives. So we, of course, need alternative solutions. And one of them is to use cultivar mixtures. So what is cultivar mixture? So basically, cultivar mixture is in contrast to monocropping. In monocropping, you have a single variety which you grow on a piece of land, and you do that every year. So you may do rotation to a different field, but you want to grow the same variety, perhaps because it is high yielding, it has other good traits which you are interested in. Now, cultivar mixture, in contrast, is where you mix varieties And the objective is to introduce heterogeneity into the field. You want to make it as diverse as possible. You want to take advantage of different profiles of resistance, different yield, so that overall you have a much more stable production. So that's cultivar mixture. But for our purpose here, we'll emphasize more on the disease part. And we know from a lot of publications, both in Denmark and outside Denmark, that if you do cultivar mixture, you have a significant disease reduction. And this is an example of this, what I'm talking about. Here I have three, on the right side, you have three different varieties planted as solo pure stands. So they are planted alone. And then on the left side, you see a mixture of them. You mix them. And you see that overall, the mixture has a lower disease if you compare to them. Overall, the mixture has a lower disease. So that's the one of the purposes we want to do a mixture, because we believe even if you don't treat, you have a lower disease epidemic development compared to pure stand, which happens in the monoculture. Now, how does cultivar mixture reduce the plant diseases? I'll go through these 5 key mechanisms. So number one, there's a dilution effect. So if you plant two or more varieties, and one is more susceptible, and the others are not, the production of spores on the susceptible plant becomes less. So because the surface area, the number of plants which are susceptible are much less in a mixture. So you have a dilution effect, which is due to the resistant population, resistant plant in your mixture. The next thing is the barrier effect. So if you have a variety which is resistant in between two varieties which are susceptible, then the spread of spores from the susceptible to the other susceptible is minimized, because you have a barrier effect caused by the resistant, which is due to the resistant variety in the mixture. You may also have compensation. So once one variety dies, the other ones will survive because of their differential or their differences in their resistant profile. So you benefit from that. There's another one called disruptive selection. In disruptive selection, because of the resistance, the profiles of resistance in the mixture are not the same. You don't have the possibility of evolution, the pathogen evolving to become either more virulent or more resistant to a fungicide or something else. So you have this disruptive selection. If you have a monoculture with homogeneity, what happens is that a pathogen will be able to evolve because they have the same substrate. So they evolve over and over again. So their evolution or selection is not disrupted. So there is a high chance of selecting a highly virulent pathogen in such system. On the other hand, where there are different resistant profiles, then a pathogen must evolve multiple virulence to be able to overcome all of them, and that becomes more difficult. We call this disruptive selection. And in connection to this mechanism, we have another mechanism called the permutation. So basically, because a pathogen, which is a pathogen is virulent to one of the components in the mixture, but not the other. Anytime it attempts to infect the one which is not, which is resistant to this pathogen, then it is possible it can induce resistance to it. So in this instance, an attempt by a pathogen which cannot attack a variety in the mixture can make the variety more resistant, because the variety becomes there is induction of resistance in this plant. So these four mechanisms, are responsible for the reason why we see this decline in disease attack in the mixtures. Now, when it comes to mixtures, are not just good for disease control; they are also good for yield, and that's the reason why many people are using them. I'll show something in my next slide to confirm this fact. So if you look at mixtures, this is data from Denmark over several years. And then we've compared mixtures, the yield of mixtures, compared to the average of the top 4 varieties, which are mostly grown by growers. When you compare mixtures to this, you see that on the average, mixtures are making more. You make more yield from mixtures. So besides you having a better disease control, mixtures give you a much more stable and higher yield. So there are several reasons why you want to do mixtures, and then in your crop IPM management. Now, because of this benefit of mixtures, you've seen a massive increase in the area of mixtures product, area that is sold to by mixtures in Denmark. If you look at the figure here, you see mixtures, which is the blue color, has been steadily increasing. And in the past 3 years, it has since 2021, it has really increased from 10% to 40% in 2024 of the area that is grown by wheat percentage. Whereas those grown by solo crop, the top 4 solo crops in Denmark, it has been relatively stable, around 31 to 20%. So you see that the benefit, the society, the growers in Denmark are seeing the relevance of mixtures due to some of the reasons I've just told you: disease reduction, and then also yield stability. So we see that there's a huge prospect in using cultivar mixtures. However, we also have another challenge, which is pesticides, which we are not able to use, or in future we cannot use. So we are trying to explore in this project how we can use alternative products. Now, overall, we've tested a lot of products. If you look at the top here, it's untreated. And then if you come down here, it's chemical reference, which is a standard fungicide. In general, we see that most of the alternative products or biological products do not give a very good effect. In most instances, they don't differ significantly from the untreated. We, however, see one product, which is a sulfur-based product, Theoprone, which gives a very good effect. So what we wanted to do in this project, this ADOPT-IPM project, was to combine this sulfur product with cultivar mixture. We know cultivar mixture can reduce the disease. In many cases, they don't prevent it, but they can reduce the disease. The biocontrol solutions or the alternative solutions can also reduce the disease. So if you combine these two, our expectation is that we can manage the disease much more effectively than when you use the two methods alone. So we did some experiments. We had a mixture made up of 3 varieties, and then we also had a solo pure stand of these varieties. We had an untreated and then fungicide reference. This is a fungicide called Balaya. And then we had Theoprone, which is the sulfur product. And then we have another treatment where we combine pesticide, a lower dose of fungicide, and we added also the biological products. How did it perform? So overall, I'm showing just 2 treatments because the other one did not really show any kind of disease, the last one. So I'm showing the fungicide reference and then the pure biological alternative product, Theoprone treatments. So overall, what we see is that if you are in a mixture, you have an averagely good performance of your against the disease. However, if we compare it to the most resistant, which is informa, we don't see so much significant difference in the mixture and then the solo stand. But if you compare the mixtures to the other 2 varieties, you see a much better effect of the mixture. A very striking effect we see here is the effect of the Theoprone, because when we see the Theoprone alone in a mixture, we see that there is a very better performance if you put the Theoprone with a mixture compared to when you use the Theoprone, for example, in pundos alone. However, if you compare it statistically across, we don't see any significant difference between them. Where we see a much more interesting result is the yield. What we see is that the highest performing, the most resistant variety, which is informa, had the lowest yield across all treatments. And then if you look at the mixture, even though the mixture did not give us the highest yield, it did not differ significantly from the best performing varieties. Now, the conclusion we get here from this, if you use a variety which is resistant alone, you may have a good disease reduction, but you may not necessarily have a very high yield. However, in the mixture, like I mentioned about compensation as a kind of mechanism, here this is about yield compensation. If you are in a mixture, overall you have a better yield because the other 2 varieties, they cushion this informa, which is not very strong, which didn't give us good yield in this particular experiment. So overall, what we see in this experiment is disease-wise, we didn't see a very marked differences. However, there was a tendency that if you have your alternative product, which is sulfur in your mixture, it does a bit better. Then in 2024, we did the same experiment again. And in this experiment, we included a variety which is very susceptible to septoria, but not in the mixture. The purpose was that we wanted to see how the disease development occur in the trial field. And then we looked at also the Bayer treatment, which is a fungicide, and then another fungicide treatment, Bayer plus Opulse. And then we had the sulfur treatments here, and then sulfur alternate sulfur with fungicide, which is the last treatment. If you look at the disease severity on the untreated, if you look at them, for example, you see that overall, mixtures were performing much better. They reduced the disease significant. There was no significant difference here, so let me not say that. We reduced the disease more than in the other treatment, except for Informa. And like I said earlier, Informa is more resistant to septoria. But if you look at mixture compared to these two, Quium and Pundus, you see there's a much better disease, lower disease in the mixtures compared to these two varieties. If you come to the fungicide treatment too, we see a similar tendency. We see that in the mixtures, you see a better performance of mixtures than all the other varieties. Now let's come to the biological, the alternative treatment. So if you look at alternative treatment also, if you see, you look at the mixture performed better than the very susceptible reference, and also better than at least one of the varieties in the mixture. It didn't perform better than the Informa, but like I said earlier, there was no statistically significant difference between the mixture and the individual components. When we alternate mixture with we alternate the thioprone, the sulfur product, with a fungicide, we see a similar tendency. We see that mixtures generally also reduce the disease. By in this case, it was the same as Informa, but a bit higher than Quium, but the difference was not significant. Overall, what we see is that if you use a mixture, the disease reduction may be moderate, but if you look at the untreated, it is really significant. It's really the difference is much bigger than when you use a product. And then if we have the thioprone alone, we see a better performance than than when you use the thioprone in a pure stand, except for Informa, which is very resistant. Then we looked at yield, the 1,000 grain weight, which is a quality parameter. And then when we look at that also, we see a similar tendency. Let's start from the untreated. If you look at the untreated, we see that there is 45.4 grams 1,000 grain weight, and which is generally higher than most of the varieties, except for Informa, which is very resistant variety. We look at the thioprone treatment. In the thioprone treatment, we see also that cultivar mixture had 44.2, which was generally higher than the reference for susceptible variety, but also for Informa over here. Once again, overall, we see that if you use a mixture, you get some slight benefits. Depending on the treatment we see, but much more when you look at the untreated, we see that it really does a lot of difference. Now, we summarize all our results we've had, not just from these particular trials we've done, but also previous years. And then we look at the improvement we get from cultivar mixtures in terms of disease severity on the first, the flag leaf and the second leaf, the yield we get in decitons per hectare, and then 1,000 grain weight, which is in gram. Then if you look at the mixtures, Quium, Informa, and then Pondus, and we compare them to their pure stance of the individual varieties, we see an average of 38% disease improvement on the flag leaf, 70% improvement on the second leaf. And then when it comes to yield, we see 8% yield gain, and then we see 3% 1,000 grain weight regarding the overall performance of mixtures only from untreated plots. So the key conclusion we've seen over the years is that, especially in treated plots, you may not see very big differences you may see some slight differences between them. Mostly, they are not statistically significant. However, you still see some benefits. But in the untreated plots, we see much more benefits. And that ranges from, for yield, for example, it ranges from, if you look at the variation, it ranges from 1 to about 3%. And then for disease severity, we can have about 10 to 38%, which you see over here. So in conclusion, what we can say is that if you have a cultivar mixture and a biopesticide, these are two very good eco-friendly solutions that we can use to control septoria in winter wheat. In fact, the fact that we didn't see marked differences between our treatments, the bio solutions, and then the chemicals suggests that this can be, in fact, a good replacement to chemical fungicide, or at least if not to replace fully, it can replace some fungicide that we use in our winter wheat plant protection program. However, it's important that we understand that for cultivar mixtures, which at this point I didn't write it here, but it's important for us to understand for cultivar mixture to be successful. It's important you compose it very well. The mixture should be made up of varieties with good profile. For example, there should be a variety which is resistant, not just only varieties which are susceptible or only varieties which are only resistant. There should be a good mixture of the components. There is also a need for you to ensure that the maturity and other physiological and morphological traits are not so different. I think in Denmark, we have a rule of the maturity shouldn't vary by more than 5 days. The plant height shouldn't vary by more than 20 centimeters. So these are some key notes that you need to always remember. You should vary the profiles of resistance. It should be made up of at least 3 varieties, and then they should not have significant differences in their morphology and then their physiological traits. So I'll end here, and I will be happy to welcome your questions.
Thank you, Isaac. Just one question about the risk of loss of sustainability of the resistant varieties. Do you think it's a risk or not? And
a risk that you lose a resistance.
Yeah.
So there's no study yet. I don't know of a study that has looked at that. But I will believe that if you have a mixture, it will rather prolong resistance gene. The reason is because, like I showed in the mechanisms, we have the disruptive selection. So if you have a disruptive selection, then you minimize the chance of evolution. So you don't have that risk of, yeah, go ahead.
And the combination with biocontrol can maintain the sustainability. Yes. Yeah, of course.
It even make it more sustainable. But the mixture itself, the concept of mixtures is supposed to also slow down evolution of resistance. So on its own, it should be able to do that. But if you add a second layer, which is, for example, a fungicide or a biosolution, then you even make it more sustainable.
Okay, thank you. We will finish now the webinar. I share my screen. Yes. So I think we are not a lot of to make the Mentimeter. I think. Do you have the link? If the people want to answer, it's possible, but we will not comment the results. So please, not the code. You can answer now because we have only 10 minutes and I want to finish now. I let the slide. You can access the site of Mentimeter with the code or directly with the URL or with the scan code. Okay. So, like I said, you can follow the progress of the project. We enter in, or we have started our last year. So you can have all the information with our social media. So if you look, you have a LinkedIn link, X, and also YouTube, where you can see some videos from trials and research topics. You can go to our website, where you can read the latest news, but you can also subscribe to our newsletter to have these news, and check out the pest management portal to see the assessment of the trials. And in the final year, there will be additional demo days. So we will have some demo days in Netherlands and in Denmark, like in other countries. We will have another round of IPE, like we did today. And we have the opportunity to invite you to our final conference. So you have to stay informed with our system, or dissemination tool, or communication tools, like social medias or newsletters, and you will have all the information. And at the end of the project, the objective is to provide you a field trials and demonstrations guide with all the results we get during the project, and as well as a learning e-toolbox with some resources. So we have the guide, some practical sheets. The idea is to provide also some e-learning material with maybe podcast. And we work also on IPM game that we developed in vegetables, on leafy, on lettuce, tomato, and we are finishing one on wheat as well. So keep informed to have all these results available for you. And thank you to attend at this meeting. And you are welcome for another one for this year. So bye-bye, everyone. And thank you for your presentations. Bye-bye.